Hi, my name is Sara Omranian. I'm currently enrolled as PhD student at Potsdam University in Germany and working at Max Planck Institute. And today I'm going to talk about PC2P, parameter-free network-based prediction of protein complexes. Today's many relational data can be mapped into network structures. One of them is biological data. And thanks to the high throughput techniques, today's millions of protein-protein interaction data are available. To analyze these protein interactions, we should use computational methods, such as clustering, which is also known as community detection. So what is considered a good clustering here is that the more connectivity inside each group, the better the clustering algorithm is. And so we really want to have less connectivity across the clusters. Our method is based on coherent partition. A coherent partition in a graph G is a partition on a vertex set V such that each partition induces a disconnected subgraph in the complement of graph G. So when we get the complement of these components, we will end up with disconnected subgraphs. As it is obvious from definition, each partition has the following properties. First, each subgraph should be a connected subgraph. As you can see, this is a disconnected subgraph. However, this one is a connected subgraph. Each component is a biclick span, and I will show you this term by examples. As you can see, this is a biclick and also biclick span. You can group the nodes into two, and you have all the possible edges between these two groups. This also is a biclick span subgraph. You can divide the nodes into two groups, and you also have all the possible edges between these two groups. And this is a biclick span subgraph again. Since you can divide the nodes into two groups, you have all possible edges between the groups, and you also have one more extra edge in one of the groups. However, this is not a biclick span subgraph since there is a missing edge. And so now I will show you this procedure with an example to see how we obtained all the clusters. Let's start with this graph, and we start with node 1. We get the second neighborhood of node 1, and then we get the complement of this second neighborhood to see if it is connected or disconnected. If it is disconnected, then we are fine and we are moving on. However, in this case, it's not disconnected, and we need to remove node or one or many nodes to make it disconnected. In this case, we only need to remove node 2, and then we obtained the induced subgraph with the remaining nodes 1, 3, 4, and 5. For this component, which is a biclick span component, we calculate the score, and which is based on the number of edges going outside to the inside, multiply 1 over the density. And then we do this procedure for each node separately, and at the end, we select the component with the minimum score, and we remove it from the graph, and again start with the rest of the nodes. In this case, each of these nodes are participate in one biclick span. Node 2 by definition is a biclick, and the other component is actually a biclick span partition. You can divide it in two groups, and you have all the edges between these two groups. So we have all the components. So after this implementation, here comes the interesting part, which is the result. So we apply this algorithm on different data sets. We use 6 data sets for yeast to human. To validate the obtained clusters, we use 2 gold standards of protein complexes for yeast, SGD and CIS 2008, and 1 for human, core. We compare our method with other state-of-the-art methods based on summation of 5 scores, namely maximum matching ratio, fraction match, separation, accuracy, and F measure. And we name it as a composite score. Due to the time limitation, I only show you part of the results. As you can see, we always have the highest maximum matching ratio and fraction match across all data sets. And PC2P also outperforms the existing methods on 75% of the analyzed data set. In human, PC2P shows the highest composite score for both PIPs and string dataset. We also analyze the performance of PC2P with respect to structural and functional quality of the resulting clusters. And for this, we determine the modularity and the geosemantic similarity of the obtained clusters across all datasets. For yeast, PC2P shows higher modularity for 3 out of 4 datasets, and for human, PC2P has the highest modularity for PIPs. In 2 other cases, MCL has the best modularity, and our algorithm is the second best with a slight difference with MCL. With respect to the functional quality of the data, the clusters, we determine the minimum and the average geosemantic similarity for every pair of proteins in each predicted complexes. So to summarize all the findings, PC2P shows the largest median geosemantic similarity with respect to molecular function in all yeast network, and it exhibits comparable distribution and median of geosemantic similarity to the other approaches. Thanks for your attention, and if you have any further question or curious about the code or the data that we use, please visit our GitHub repository.